Hello everyone, welcome to the new session of Mendel Laws of Inheritance. So this is the second part for this particular topic. In this topic, we are going to study about the laws of segregation. That is the second law of the Mendel, right? So this law is also called as a purity of gametes. Purity of gametes. So let us get into the details of this particular law, right? So the law of segregation states that when a pair of contrasting factor or genes or allelomorph are brought together in a heterozygote, that is a hybrid, and the two members of the allelic pair remain together without being contaminated. When the gametes are formed from the hybrid, means the hybrid which is formed in the F1 generation, okay? So the hybrid form the two separate out from each other and one enter each gametes. So let us understand this particular explanation of the laws of segregation with the example, right? So let us take an example. So I'm taking an example of a tall plant of P, right? So I'm denoting capital T as a tall plant and small t as a dwarf plant. So this is dwarf plant and this one is the tall plant, right? So one will be a male and one will be the female. This is the parent. This is the parents, right? Now there will be a crossing in between and we will get capital T, small t, right? Because only the two types of gametes are formed in here. This is capital T. Here it will be small t. So only one type of gamete, that is capital T, from the male, and only one type of gametes from the female, that will be the small t, and they unite to form capital T, small t. This is the F1 generation, and this is the hybrid tall. So if we see the explanation, that what does the explanation says for the law of segregation, that the members of the allelic pair remain together without being contaminated when the gametes are formed from the hybrid. So they remain together. These two alleles, that is one allele for tall plant is capital T and another allele for the tall plant is small t. They are together. Right? As a hybrid tall. Now, what happened when they form the gametes? So here in this case, they will form two types of gametes. So again, I'm taking two plant, capital T, small t, capital T, small t. One will work as a male and another will be the female. So they will produce this time two types of gamete, capital T, small t, capital T, small t, right? In the first generation, both are homozygous. This was the homozygous tall and this was the homozygous dwarf. So they produces only one type of gamete, that is the capital T and small t. But here in the case of both the parent, male and female, are heterozygous tall. So they produces two types of gametes, capital T and small t. Okay, so this also produces capital T, small t, right? Now what happened? The capital T will unite with the capital T. So this can be a one of the combination, that is capital T and capital T, right? Another combination can be capital T and small t, capital T and small t. And this can also be a combination, small t and capital T and small t, small t. So here we can see that in the F1 generation, the two alleles or allelomorph right? That is the capital T and small t are together, right? But at the time of gamete formation, both the alleles are segregated. Both the alleles are segregated, right? And this is the purity of gametes. Like the gametes are pure at this particular condition. The gametes are pure. Capital T, small t, capital T, small t. There is no mixing in the case of gamete formation at the F2 level, or the F2 generation, right? So that is why it is also called as the purity of gametes. Okay. So pure tall plants are homozygous. So again, this is the discussion for the cross which we have discussed just now, right? So pure tall plants are the homozygous and therefore possesses genes that is also called as factors by the Mendels. Later it was called gene. So the capital TT, similarly dwarf possesses genes small tt, right? The tallness and dwarfness are the two independent but contrasting factor or determiner. So the capital T and small t both are contrasting. The capital T is for the tallness, right? And small t is for the dwarfness. So both are totally a contrasting character. So at the F1 generation, what happened? Both the alleles are together. And that is why it is called as hybrid. And if we see the phenotype, it will be totally like a tall plant. So it is called as tall hybrid. But at the time of gamete formation, they will form two types of gamete, capital T and small t, right? So two types of gamete, and that is why it is called as purity of gametes. Okay. So, pure tall plants produces gametes, all of which possesses T, and dwarf plant possesses T types of gametes. So, in the F1 means when the parent are there, the capital T and capital T, the small t and small t, they will produce only one type of gamete, that is the capital T, and this will produce only one type of gamete, that is the small t, right? So, during cross fertilization, the gametes with T and capital T and small t unite to produce hybrid of F1 generation. So here they will form a hybrid, capital T and small t. Both the alleles are contrasting, but they are together in the F1 generation. So these hybrid possesses the genotype capital T and small t, right? So this is the genotype. Genotype is capital T and small t. But what will be the phenotype? Phenotype will be tall hybrid. Actually, we cannot differentiate the tall plant from the genotype capital TT or the tall plant from the capital T small t. Phenotypically, they will look alike, but when we see the genotype, so this one is the hybrid one. Okay. Next, it means F1 plants, though tall phenotypically, possesses one gene for the tallness and one gene for the dwarfness, right? So capital T, small t will give one type of gametes only, capital T, and this will small t. They unite together. Fertilization take place in between, and capital T, small t type of tall plant will be formed, and this is hybrid. So it is said that it means F1 plant, though tall phenotypically, possesses one gene for tallness and one gene for the dwarfness. Okay, so apparently the tall and dwarf characters appear to have become contaminated, developing only tall characters. So the capital T is the dominant gene, the dominant allele, and small t is the recessive one. So it masks the effect of the small t, that is the dwarfness, right? So here we can see there is a contamination. There is a contamination of the gametes. Okay. Actually, it is a contamination of the allele. But at the time of gamete formation, the gene T for the tallness and small t for the dwarfness separates and are passed on to the separate gametes. Now, what happened at this particular level, that is the F1 level or F1 generation, this plant will form two types of gametes, that is the capital T and small t, capital T and small t. So now there is no contamination at this particular level. The gametes are totally pure one. The capital T, that is the gamete for the tall, right? And small t, that is the gamete for the dwarf. Okay. Okay, so this is the Mendel laws. We are discussing the Mendel laws. So again, we are coming to that particular cross. This is the cross for the Mendel's second law, that is the laws of segregation or the purity of gametes. So Mendel took a pea plant, that is capital T, and the dwarf, that is small t, plant, made a cross between them. This we have already discussed. So tall and dwarf plant, this is the capital T, and this is the small t. So what will happen? Small t and capital T plant will be formed. That will be hybrid tall, right? Now we will take two plants, one acting as a male and one acting as a female, right? So in the first generation, he got the result as a dominant tall plant. So all plants are dominant tall. This is the dominant tall, but this is actually a hybrid one. The Mendel self-fertilized the F1 generation and obtained 3:1 ratio. So here he got 3:1 ratio. 3 tall and 1 dwarf. 3 tall and 1 dwarf. He correctly theorized that the genes can be paired in 3 different ways for each trait. So here we can see capital T and capital T, small t, capital T, small t, capital T and small t. So two types of gametes, capital T, small t, capital T and small t, right? So three types of pairing can be, we can see capital T, capital T, small t. Again, this will be capital T and small t, but this will be small t and small t, right? So these two are pure homozygous form, that is the tall, pure tall, and this will be pure dwarf, right? And this will be hybrid tall. So this is capital T, small t, and capital T, small t type of three types of plants we are getting at this F2 generation. But this is because formation of pure gametes at the F2 level. So as a result, two types of gametes are produced from the heterozygote in equal numerosity. So two types of gametes. We have just seen that the capital T and small t will form two types of gamete, capital T and small t, and equal in number. So that is why it is said that heterozygote in equal numerosity. So capital T, small t are equal in number, and this was the heterozygote that produces equal number of gamete, but two types of gametes. So 50% of the gamete possesses the gene T and the 50% possesses small t. Therefore, these gametes are either pure for the tallness or dwarfness. That is why the law of segregation is also described as law of purity of gametes. Okay. So here we can see that capital T, this can be a male plant, this can be a female plant, right? So capital T is type of gamete, small t of type of gamete. The female will also form the same, that is 50% for the tallness. The gamete is having 50% for the tallness and 50% for the dwarfness gene in the gametes. Okay, so 50% this one, 50% this one in the case of female, and again in the case of male, 50% capital T and 50% small t. So the gametes are pure. Gametes are not in a heterozygous form as the parents at the F1 level were. Okay, these are not pure form, they are contaminated. One allele is capital T and another one is the small t, right? But if we see at the gamete, the gametes are totally pure, and that is why it is called as purity of gametes. So the gametes unite at random. When the gametes are numerous, all possible combination can occur. So there can be a number of combination. We have seen this, how many type of combination these gametes can form. So with the result of that, tall dwarf appear in the ratio of 3:1. Okay, just now we have seen, and the results are often represented by the Punnett score, that is as follows. So let us see this. So it has been confirmed by the cytological studies that the dominance or number of dominance, the law of segregation holds good to all cases. So cytological studies confirm that the dominance or number of dominance, the law of segregation hold good to all cases, right? So its far-reaching applicability has made it rare biological generalization. So this was the generalization for all type of plant, and that is why it becomes a law, that is the purity of gametes. So the critical appreciation of the law of segregation, if we take an example, that is the capital R, small r at the parent level, then they have one gene for round. So this is for round, right? And small r for the wrinkled, right? So capital R or small r capital R have gene for the round and wrinkle. Okay, so they will have only wrinkle gene, right? So RR, capital R, or small r capital R, these are having round and wrinkled, but this will appear as a round gene, round seed, right? This is pure round and this is pure wrinkled. So what happened at the time of gamete formation? This will form capital R type of gene and this will form capital R and small r, two type of gametes, capital R and small r, right? And this one will have capital R. So what happened? There will be a combination of capital R. There will be the combination of capital R, small r. There will be the combination of capital R, small capital R, and there will be the combination of capital R and small r. So we are having two pure round and two hybrid round. Okay, so here we can see capital R, capital R, small r, small r, and this will be capital R, small r. So phenotypic ratio will be round wrinkled are 3. This is round wrinkled. Okay, and this one will be round. So this is the ratio. Okay, so we are taking one more example to understand this. Pure flowers. Okay, and this is the white flowers. So what we see in the F1 generation, all plants have the purple flower. So if the purple is denoted here as P, so this will be the condition. And at this, we will have P small p. That is the hybrid. Hybrid. Right? All plants have the purple flowers. Fertilization among the F1 plants, that is the F1 into F1. Right? We will take one plant as a male and one plant as a female. So this we can see with an example, like we are taking capital P and capital P, small p as a male plant and this one as a female. So it will form two types of gamete, capital P and small p, capital P and small p, right? And they will unite in this way. There may be different combinations. So one of the combination can be capital P, capital P. This can be capital P, small p. This can be capital P, small p. But this one will small p, small p. So these three are purple flowers and this one will be the white flower. So 3/4 of the plants have the purple flowers and 1/4 of the plant will have the white flowers. So this it has been shown here. The phenotypic ratio is 3 purple and 1 white, and the genotypic ratio will be 1 pure purple, 2 hybrid purple and 1 white flower. Right? So this is about the law of segregation of the purity of gametes. Okay, so this is all about this session. We will meet with one more session to discuss about the Mendel's third law, that is the Mendel law of independent assortment. Okay, till then, thank you very much.